Okay, so on November 9th, which is I'm recording this, was yesterday, Pfizer and BioNTech announced that they had a vaccine candidate against COVID-19. And so I am going to go through some of the biochemistry of COVID-19 and talk about how this vaccine, which is called uh, BNT162b2. So I'm going to go through some of the background biochemistry on COVID-19 and then talk about how this vaccine is supposed to work. And so uh, SARS-CoV-2, which is the name of the virus, um, which is oftentimes called maybe coronavirus. So you hear that a lot in the news, or just there should be an O, COVID, or COVID-19. And so these are all things that you hear quite often in the media. You don't hear it as often, but I remember back when this first started, they sometimes called it the novel coronavirus. And so the coronavirus, or COVID, is a beta coronavirus, which is the genus that it's in. And so if you remember from your biology 101, there's kingdom, phylum, class, order, family, genus, species. So this is the taxonomy here of COVID-19, of SARS-CoV-2, and it's quite related to MERS, which is the Middle Eastern respiratory syndrome, where SARS stands for severe acute respiratory syndrome. They have that down here for species. So severe acute respiratory syndrome. This was SARS from back in the early 2000s, what back then was just called SARS. And so you can see how that's branched off here from SARS, and then how MERS and SARS-CoV-2 branched off here. And so to look at the sort of the genomic structure here of SARS-CoV-2, what we see here is that SARS-CoV-2, its genome is a single strand of RNA, and so that's its genomic structure, and it has these two open reading frames here. That's what ORF stands for, open reading frame. And what's interesting about that is the open reading frame. So there's this overlap here, and so what actually happens is from here all the way to here can be translated in one reading frame, or it can translate all the way up to here in one reading frame. Then there's this slippage site. There's this frame shift site where then it can move over and start translating this part in a different reading frame, and so you actually get multiple different proteins from the same stretch of RNA here, and so we can get these multiple different proteins. And so down here I actually have, these are the two different open reading frames. So if the virus is being translated in the first open reading frame, then it translates this entire area here, which generates a polyprotein. So something like this. It's a number of different proteins that are connected together, which then later on, at these places where the arrows point, will be cleaved into multiple separate proteins. So the ORF1A first translates all of this as a single large sort of what's called a polyprotein. And if you translate the other open reading frame, then you get these first few proteins are the same up to this NSP 10, and then you switch to that other open reading frame, and then you also generate these proteins here, which are then cleaved by things like this PL proteinase and this 3CL. Those are both enzymes that cleave proteins at these particular sites. And so up here, I actually have the genomic structure of several other coronaviruses. And so the OC43, the HKU1, the NL63, and the 229E, those are all coronaviruses that are involved in certain strains of the common cold. So the common cold can be several different types of viruses, with coronaviruses being one particular type. And these four are coronaviruses that can cause the common cold. And so we can see that they have a very similar genomic structure here, or genomic pattern, I guess you could call it, to the SARS coronavirus. And so that's kind of how you identify genomically that something is a coronavirus. So yeah, like I said, it generates this polyprotein, and these. So this is all just from the open reading frame 1A and 1B. Then we also have this right here, which is the spike protein, which we will be talking about in more detail later, because this is the protein that's actually involved in the virus getting into human cells and infecting them. And so the way that actually happens is, so we have the SARS-CoV-2, we have our spike protein, which are these represented by these blue things here, and they bind to the receptor ACE2, which is a protein in human lung cells, amongst other types of cells. But for the SARS-CoV-2, the lung cells are where it's primarily infecting. And so this allows the viral cell to be brought into the cell, so that's entry. And then the virion, which is what the viral cell, I mean, it's not really a cell. Viruses are not really cells in the way that we understand them, but the virion will release its RNA, will release this RNA up here into the human host cell, which these are then transcribed, translated rather. So this is already an mRNA, so it's translated into proteins. These are our two polyproteins here. They go through the proteolysis, which is the process of breaking them up into their individual proteins. And then those form this complex called a replicase, which then can replicate the mRNA, the viral genome, and so you can generate multiple versions of the viral genome. And the mRNAs from that can also be translated. So this S here, once again, is the spike protein we'll be talking about soon. And these are actually brought into the endoplasmic reticulum, and then they move into the Golgi apparatus for further processing. And then the replicated genome that we replicated from our other proteins, from these other proteins that were split into their own individual proteins, can all be packaged once again into this virion, and then you can have multiple viruses being released. And this figure here actually shows some various types of antivirals, and where in the viral life cycle they are actually attempting to stop this infection. Stopping at this replication is where things like, so you may have heard of remdesivir as a potential drug to use against coronavirus, and that stops here at this replication stage here. But then we can also have drugs that attempt to try and stop the entry of the virus into the cell in the first place, and we can also target the host enzymes. And so this process here of going through the endoplasmic reticulum to the Golgi apparatus will hopefully be inhibited so that the virus cannot reassemble itself and keep reproducing. And so that's sort of the way that the SARS-CoV enters the cell. And so let's get into this process here a little bit, because that's the background we'll need in order to understand how the vaccine works. And so here again, well, this is the spike protein, and so the spike protein can be divided into two sections. So there's the receptor binding and the membrane fusion sections, and these sections of the protein are actually split by a human protease called furin. And so this furin binding site, or this furin cleavage site here at the S1, S2 cleavage site, so we can see the sequence here of that furin cleavage site. And what we should notice is this hasn't highlighted these R's, which are arginines, which are an amino acid residue in the protein. And these are positively charged amino acid residues. And so these positively charged amino acid residues, which K, which is lysine, is also a positively charged amino acid. And so this positively charged site here is where the cleavage is occurring in the spike protein. So down here we have the furin recognition motif. Once again, you can see all these arginines here. And so this insertion here, this PRRA insertion here, is actually what identifies the SARS-CoV-2 furin motif. And so it's this insertion that scientists think is what caused the SARS-CoV-2 to jump from the bat and the pangolin versions to humans. What made it jump species from these bat and pangolin species to the human species. And so this cleavage site gives it a different binding to our furin protease, so that it can actually be recognized by the human version of that enzyme. Up here we also have between the human, the pangolin, and the bat, we see that we have these conserved amino acid residues in the spike protein. These are the amino acid residues that actually bind to the human ACE2 receptor. These are well, I guess not just human, it would also be pangolin and bat, but these are conserved residues that are involved in that binding. And so we can actually see that. I have the crystal structure here of the SARS-CoV-2 spike protein, and I have the PDB codes and the paper references in the description down below if you're interested in looking more deeply into this. And so we can see, as I was talking about before, we have the receptor binding and the membrane fusion domains, and I have the receptor binding domain in the cyan color here, and the fusion domain in the magenta color here. And so we can now also see other different domains here. So there is the HR1 domain, which we'll get into more in a little bit, which I have in this sort of ruby color here. And I have up here in the more silver color the parts of the receptor binding domain that actually interact with the ACE2 receptor. And I have in stick version here these amino acid residues that actually interact with the ACE2 receptor. And what you can also see here is that so the spike protein is what's called a homotrimer, and it's called homo because they're all the same, and it's trimer because there are 3 different proteins involved. So if we look down at this, we can see sort of the 3 proteins coming together into this triangle. But what we also see is that this one right here and this one right here are in what's called the closed conformation, where this one here is in the open conformation. And the open conformation is needed in order for this to bind to the human ACE2 receptor. And so we can see this structure having 2 in the closed and 1 in the open. And then between these 2 sections here, between the cyan and the magenta, there will be the section which we can sort of zoom in here. So this actually has a loop that would come around. There were some amino acids that they weren't able to model into their structure because they were too disordered. But this is the place where right here the S1 or receptor binding part of the protein turns into the fusion part of the protein. And it would be the sequence here which would have those arginines that would actually be cleaved, and that will release the S1 domain from the S2 domain. And we'll take a look here soon at what happens there, because that causes a rather large conformational change in the S2 domain, in the membrane fusion domain. And in fact, we'll take a look at that right now. So once again, we have our coronavirus here. You can see it has the RNA inside, the S on here, these purple things. And we can see this split up into the S1, which contains the receptor binding domain, or RBD, and S2, which contains the fusion domain. So the FP is the fusion peptide. And then there are these HR1 and HR2, which are heptad repeat domains. And what happens is we have our protein here, our spike protein, which is bound to the viral membrane. Then there's the ACE2 receptor, which is bound to the human membrane. And so we see this is in the down position, and then one of them goes to the up, like we saw in this, where one of them is now in the up position, and that allows it to bind to the human ACE2 receptor. And then we will have the cleavage, and that happens from the furin enzyme. And what that does is, so now they show it no longer in this in the protein structure form, but in this sort of cartoon form, is these S1, which is which is the part of the protein that contains the receptor binding domain, comes off of the spike protein, which then has this large conformational change. And I didn't like the way this showed it, so I liked down here. So down here we have our spike protein. So this actually shows the spike protein on top. So up here, this would be our viral membrane, and down here would be our human membrane. And so we see this comes down. It's in the down position. It moves to the up position, binds to the ACE2 receptor here. Then there's the protease action, which causes these S1 domains to break off, so that we end up with just this S2 here, which then undergoes this conformational change. It spreads out. This R, the fusion peptide, it now binds to the human cell membrane, and now it starts bending over, which brings the two membranes close together, which this is going to a different step. It sort of clamped them together, and now we are combining our human and our viral membranes, so that, like we saw up here, then we can actually start bringing the virion into the human cell. And so, like I said, this spike protein undergoes a pretty large conformational change once the S1 domain is cleaved off. And we can look at that right here. So I actually have here. So this is the top of the S2 here. We can look down at it, and we see that it forms this triangular sort of structure here, and they actually show that right here. And so we have our HR1 forming this triangular structure, and then our HR2 sort of on the outside, and we can see that right here where these I guess olive colored ones in the middle here are our HR1, and then it's kind of hard to see. Maybe if I switch over like this, these ones here would be our HR2. So I can actually highlight the HR1 here, and we see that that's stretched out. This is in the what's called the post-fusion form of the protein. So I have that highlighted with the little dots here. And that's the HR1 in the post-fusion in its stretched out version. Now if I highlight where the HR1 is in the pre-fusion, you see it's down here and it's sort of folded back in. So what happens is these blue parts right here actually extend out so that it becomes like this. And they don't have it modeled on here, but up here is where the fusion peptide would be. Those were likely too disordered to get a good structure for them. But coming off on the top of the HR1 up here would be the fusion protein, or the fusion peptide rather. And so we see this undergoes this large change where these HR1s, so the HR1s here in the pre-fusion conformation are sort of tucked down this way, and then once that large S1 head comes off, so we can see here in the ruby color is actually the HR1 on here in the pre-fusion conformation. And so we see once this cyan part comes off after being cleaved, then this part will stretch out and become this right here. And so that's how that's what we see happening here, where this is in the sort of pre-fusion conformation. Then we see it stretching out with our fusion peptides, grabbing onto the cell membrane and sort of squeezing these together so that the virion can enter the human cell. And so the spike protein is the target of the vaccine. And so this vaccine, instead of doing what a lot of vaccines do and sort of just throwing in a piece of the protein, so for in this case we might send in a piece of the S1 protein and then just inject that into the body, or in some sort of attenuated virus, so that the human immune system, so that these antibodies like this would grab onto it and then signal to the T cells that we need to look for this protein structure here and attack anything that has that protein structure on top of it. But in what Pfizer is doing here is actually an RNA version of it, so that they're actually injecting this RNA, which they are calling the BNT162b2. So it's an mRNA, and they actually put that into the human body, and then the mRNAs will be taken up here by the white blood cells. So these are the mRNA delivery vehicles, which are these little nanolipid vehicles here that have the mRNA inside. And so the white blood cells will find these, they'll take them up, they'll recognize this as an mRNA using these endosomal sensing, which then these will start telling the white blood cell to start producing these interferons, which are our body's immune chemicals against viruses. And it will also use these mRNAs to actually translate the part of the S1 protein here as an antigen, and it will then put this onto the tips of these MHCs, which are called major histocompatibility complexes. And then they actually sort of present these on the surface of the cell, so that other white blood cells can come and sort of grab onto it and say, okay, those are the things we need to look for, and we need to start producing more of these kind of cells. Now, that's also what the interferons do as well. And so these mRNAs are produced in vitro. So this just shows that we're using these DNA circles here. They're called plasmids. So they produce the DNA. They transcribe it in vitro. Then they use some various enzymes to actually process the mRNA. So they add the 5' G cap and the poly A tail, as well as these untranslated regions, which are actually what, well, this is the UTRs are the untranslated regions, and those actually control the translation of the mRNA. So, like I said, these mRNAs that are being produced will be injected into humans, and then it will be taken up by the white blood cells, and this immune response will proceed. And so these are the structures of the antigen that Pfizer. This is actually a figure from the paper that is has recently come out. These were generated by cryo-electron microscopy, and so I couldn't find these structures on the protein data bank or on the EMBO cryo-electron microscopy database or anything like that. So I couldn't compare it directly with this, for instance. But we can kind of see this has this sort of this bulged top shape, and so does this. So we can sort of just at a first glance see that these are similar in structure to our spike S1 protein, which is what this this mRNA, this BNT162b2 mRNA is actually coding for. And so that gives us our antigen, which the white blood cells can then present on their surface on the MHCs, the major histocompatibility complexes, for other white blood cells to then sense and start mobilizing an immune response. And so this is also from the paper. And so what they were looking at is the receptor binding domain and IgG response. And so we can look down here at this key. So the Rbd binding IgG response obtained 7, 14, 21, 28 days after immunization, determined by ELISA. And so this is looking at the so the IgG is actually what they are trying to get the cells to produce. This is the antibody that they want the cell to produce. And so by presenting this antigen here, that will tell other cells, like these T cells, to actually produce that IgG antigen. And so they want to look at the IgG that can actually bind the RBD, the receptor binding domain of the S1 protein. And they can do that using a technique called ELISA, which I actually have over here. So I don't think this is exactly the ELISA technique they used, but it's ELISA are all similar in that you have your capture antibody, and then you put your antigen on it so that it's stuck to the surface, usually in like something like a 96 well plate or something, and then you have your biotin labeled detection antibody. So this down here would be the antibody. This would be the IgG that they want to test, and then they have this that binds to it, and then you bind this enzyme to it, and then you can actually look at the production of colored substrate, and you can look at how much of this substrate is being produced, and it will only be produced if your antigen has bound, because otherwise this whole apparatus will not bind to it, and so you wash it away, and then you get none of this product being produced. And so that's how this ELISA assay works. And so they're looking at different amounts of the BNT162b2 being added. The 0, which you can see is actually down here, and that's what you would expect, that you would get no response. And so here we actually get response after the 7, 14, 21, and 28 days. And so that is that's promising there. And then over here we have we have the plasmon resonance assay here, and that is looking at the binding affinity of the so they're looking at the binding affinity of their IgG from the serum after 28 days after immunization, and they're trying to bind it to the to the S1, which is the full protein, or the receptor binding domain, which is just a section of the protein. And so a surface plasmon resonance is done like this. And so so we can see here, we actually bind our our protein onto this surface, and then we can have our IgG here. I think in this paper they actually had the IgG bound to the surface, and then they were washing protein over it. But the principle is the same, and so then you actually shine this light on it, and depending on how much bound versus unbound protein, so we have our unbound here and we have a bound here, will change actually the angle at which the light reflects off of this surface. It actually changes the conductance of this surface. And so you can look at how much the angle of reflection changes. And with that, you can measure. So this is actually measuring the resonance. This is actually measuring how much the angle has changed. And we can see here, I believe these are at different concentrations. But anyway, you can see here that when they start washing the protein onto it, you see the angle changing as you start binding, and then as you stop washing protein onto it and just start washing it with the solvent, your binding starts going down. But we can see here that the KD, which is the binding affinity, is at 1.2 times 10 to the negative 8 molar, and so that is on the order of nanomolar affinity, and so we start getting a lot of binding even when we're at extremely small concentrations, and that tells you that it is a tight binding between the IgG from the immune response and the S1, which is on the left here, and the receptor binding domain, which is on the right here. And then over here, what they actually have is these pseudo virus neutralization assays. And so what they have on the x-axis down here is actually the dilution, so it's a serial dilution, which is why it doubles from 8 to 16, 32, 64, 128. And we can see with a low concentration, intermediate concentration, and a high concentration of their RNA, that you actually see that these cells are gaining this immunity in a dose dependent manner. And I actually have the cell neutralization, this pseudo virus neutralization assay up here. And so what they do there is they actually use plasmids with the proteins of interest and put it into a different kind of virus, one that's easier to work with and perhaps less dangerous. And then they can actually look at when they infect cells with this virus, they can look at the infection rate of the cells. And if we get infection, so they're actually looking for fluorescence in the cell. Fluorescence, I think that's how you spell it. And so you can you can measure to see how much fluorescence is in the cell, and if you have no fluorescence, then your cells are not being infected, and so that's how you know that the virus has been neutralized. And so that's what they're doing here at different concentrations with a serial dilution. of the mouse serum. And so what they did is they immunized the mouse with this amount of the RNA, and then they took serum, they took samples from the mouse, then they diluted it. They did this serial dilution and looked at whether the cells in those dilutions were able to neutralize the virus. Uh, and by the way, once again, these figures that I'm looking at here, so this one here, and now this one that we're going to look at, all come from the paper that the scientists released, which is this one, and I have it linked to in the description below. It's a prefusion SARS-CoV-2 spike RNA vaccine is highly immunogenic and prevents lung infection in non-human primates. And so I do believe that they have been doing human trials. So this one was posted on September 8, but this is the one that where they were doing the non-human and in vitro trials with this particular vaccine. And so this is looking at the receptor affinity. Well, first it's looking at the expression, and so we can see up here the expression of the IgG, I believe. No, it's the P2S. So that this is actually the expression of their antigen that they're looking at here. And so, as you would expect, the control doesn't express any of it, because why would it if you didn't put any of it in? And these two are expressing the antigen, which was that S1 protein. And so down here, we're actually, so this was measured by flow cytometry, which is actually this process here. So you have your cells. The antigen in these cells is actually labeled with another fluorescence, and so you can measure as your cells move through this really tiny tube, one cell at a time. You are shining a laser on it and actually measuring the amount of fluorescence. And so that's what they were measuring in that part of that figure there, was the fluorescence of the cells from flow cytometry. Then the B and C here, the P2S with C terminal twin strep tag. So this tag is probably what they use to actually purify the protein. It's expressed in this line of cells, and was solubilized, blah blah blah, was assayed with biolayer interferometry. I think it's interferometry. The binding kinetics was immobilized with the ACE2 and the B3 mononucleal antibody. And so this is similar to that surface plasmon resonance, except instead of looking at the angle that you're changing when proteins bind to other proteins affixed to the surface, you're actually measuring the phase change here as proteins bind to your surface. So you're measuring this change in the phase here, or actually I think this is in the actual wavelength. But anyway, it's a similar technique to the surface plasmon resonance in that you are using the change in the change in the surface, in the conductance of the surface, to actually measure the light coming off of it. And so once again we get our KD which is in the nanomolar range here. So it's very tight binding. So the P2S binding to the ACE2, and so this is looking at their antigen binding to the human ACE2, and this is looking at it binding to this B38, which is a monoclonal antibody. Okay, then up here we actually, once again, this is still from that same paper. These ones right here. So we have our S1 specific IgG. So this is just the levels of IgG in the days after the first vaccination. So this on the left here is with a 30 microgram vaccination. This over here is with a 100 microgram vaccination. Over here we have our neutralization assay. And so, like I said, that's this one right here, where we're looking at whether the cells are actually neutralizing the virus. And so that's what this VNT50 here. I think that's something like 50% of cells are neutralizing the virus. Down here we have our peripheral blood mononucleal cells, which are I have listed over here. So those are the monocytes and lymphocytes and things like that. And so we're looking at the IFN, which is the interferon gamma levels, with a 0 microgram inoculation, which we see down here. We get no response. 30 micrograms and 100 micrograms, we see that after 14 days we start getting a small response. 28 days we see this peak response. And then this is for the interleukin 4. And it's looking at the same thing, so we start seeing this response, but it's a bit less of a response for the interleukin 4. Down here we have the CD4 T cells that are expressing the interferon gamma, the interleukin 2, and the TNF alpha, which is tumor necrosis factor alpha. And we can see that we start getting some more of those, these CD4 T cells. And the interleukin 4 once again seems to have a much more depressed response. We're getting barely anything in our experimentals here. The interferon gamma CD8 T cells. We start seeing some response there, particularly after about 28 days. And over here, I just have copied from Wikipedia just what the interferon gamma does, and that does things like promote natural killer cell activity. Natural killer cells being one of our PBMCs. It's a type of white blood cell. Increases antigen presentation, which is what we are trying to do with this S1, is have our vaccine, which is an mRNA, start producing this antigen. And so if our immune cells, which we saw down here, start releasing these interferons, then we will start having more antigen presentation. And so that is why we want that to be something that we see here. It does a few other things, but those are the ones that I'm going to talk about here. Down here we have our TNF alpha, our tumor necrosis factor, which is a potent chemoattractant of neutrophils. So it brings white blood cells uh, also stimulates phagocytosis. So it's stimulating the immune system, which is also something we want, and that's why it's over here, TNF alpha. That's why it's good that we see this response over here. And the interleukin 4 is a cytokine that induces differentiation of naive T cells. So this is what will make our T cells turn into T cells that specifically respond to this antigen. Unfortunately, they seem to have only very little of the IL-4. We seem to have some IL-2, which is a different kind of interleukin, a different kind of cytokine, but little of this IL-4 response. Up here I just have the anatomy of the IgG, which is that antibody that we want, and the way they work is they're in this Y shape, and they have this section here in the branches of the Y that are quite variable. So the amino acid sequence of them are quite variable, and so what happens is we have a bunch of different variations of it sort of floating around in our bloodstream, and when they connects with something, when our if this is our antigen, if this is our S1 antigen, if one of these happens to bind the S1 antigen, then what this does is at this end it sort of signals to white blood cells, hey, I've bound onto something. This is something that shouldn't be here. Start making more of me. And so that's why we want to get this IgG response. We want to start seeing this increase in the IgG, and this is the binding affinity that I was talking about up here, where this is binding to the S1. We want it to bind tight to the S1, and we want it to bind specific to the S1. We don't want this binding to things, particularly things that we want in our blood, and which would cause an allergic or autoimmune response. So we want this to be specific, and we want it to be a tight binder to our S1 here. And then also what these do is as they bind to cells. So if we have our virus here and it has its S1 coming off, if this binds onto the virus, then it starts blocking the virus from entering cells. These will also bind to other IgGs that are bound to viruses. And so these will bind, and then we get what's called agglutination. I think this is how it's spelled, agglutination. And that just sort of turns this into a kind of a big blob that is unable to infect cells. And so not only do these antibodies sort of tell the immune system to start ramping up, to start mobilizing an immune response, they can also agglutinate these viral particles and neutralize those. So that's what that neutralization is. If we start getting a lot of these binding to the viral cells, they cannot enter the cell, and so we don't see this fluorescence in the cell, and that's how we know that it's neutralized. This it had so here is just different strains of the SARS-CoV-2 virus here. So these are just different mutants that have been found, and you can see the flags for the country of origin. A lot of them are in China. A lot of them are in the United States. And so these are the bat CoV, and this is the human SARS from back in 2002, and the MERS here. Once again, this is that frame shift. Um, these are the genomes of the SARS-CoV-2, the Pangolin CoV, and the bat CoV. You can see that there's a lot of overlap there. All right, this is what I wanted to check out. All right, so these are looking at the side effects of this BNT162. Well, this is actually B1. Down here is the B2, and so this is looking at percent of participants who had pain at the injection site. So that seems to be pretty high. Redness seems to be pretty low. Swelling, pretty low. And so this is looking at different concentrations of the mRNA of the vaccine. We see pain at the injection site had very little with placebo, and so that pretty much tells us that that this does cause some pain at the injection site. There's very little redness, very little swelling. So this is dose 2. I guess I should have pointed out this vaccine has to come in 2 doses. So you have to get a first dose and then go back later to get a second dose. So this is looking at second doses. And the BNT162B1 was just another form of this vaccine that they were looking at. And you can see here that the difference between the B1 and the B2 version, there's a bit more swelling in the B1 version. There's a bit more swelling here. And so that's why they went with the B2, because it had less of these negative effects. Then we can look at this over here, which is different side effects. So fever, fatigue, and chills. So this seems to be a bit higher in the B1 than the B2, but there seems to be little fever. At the high dose, we get a little bit of fever. The high dose here, we get a little bit of the chills. There does seem to be a bit of fatigue going on here in the first dose. These seem to be increased in the second dose. Down here, so the top are actually participants of 18 to 55 years of age. The bottom ones are participants of 65 to 85 years of age. And so the older participants, once again, the B1 version seem to have more side effects than the B2, which is likely why the B2 was chosen over the B1. But the older participants don't seem to have these side effects as bad as the younger participants, who particularly had it in the second dose. Yeah, and so that is the vaccine that is being worked on as we speak, or at least as I speak right now while recording this. I don't know when you are watching it. And so that is the this promising RNA vaccine. And like I said, this paper, amongst other ones where I grabbed these figures here from, are all linked to in the description down below. And so, yeah, that is the vaccine. There was actually one other thing I wanted to just quickly. So this is actually a post on my blog that I wrote back on March 13, talking about the coronavirus. And one thing I just wanted to quick read, because you don't hear that much about the ACE2 receptor, which seems like it's kind of a strange one or it's just kind of like, why is it the ACE2 receptor that's being bound? The ACE2 receptor, I'll just read from what I wrote back in March. The normal physiological function of ACE2 is in the processing of angiotensin. And then I just have here the etymological root of it, angio, as in the heart. Think angina and angiogenesis, and tensin, as in causes tenseness. Uh, so the ACE2 is in the processing of angiotensin, a peptide hormone that controls vasoconstriction, tenseness of the blood vessel, and blood pressure. Think ACE inhibitors for people with high blood pressure. ACE2 is a type 1 membrane protein expressed in lungs, heart, kidneys, and intestine. Decreased expression of ACE2 is associated with cardiovascular disease. The peptidase domain in the heart, the part of the protein that does the processing of angiotensin of ACE2 cleaves angiotensin Ang1 to produce Ang1-9, which is then processed by other enzymes to become Ang1-7. ACE2 can also directly process Ang2 to give Ang1-7. This causes blood pressure to increase. So the takeaway is that ACE2 is in charge of this vasoconstriction process here. And you, like I said, you may have heard of ACE inhibitors, and so that's what the ACE2 is. And I just kind of wanted to point that out, why we got this. Actually, this is another thing I wanted to show. This is actually the SARS-CoV from back in 2002 spike protein. You can see it's a very similar shape. And so this is actually the SARS-CoV-1 spike protein. And this up here in magenta is the receptor, or is the ACE2 receptor, or at least part of it right here. And we can see that this open part of the spike protein here is bound to the ACE2 receptor up here. And once again, the mechanism for this would be very similar to the SARS-CoV-2 that we are dealing with right now. And so, yeah, this is how it binds to the ACE2 receptor up here. And that was what I was talking about over here, where this is the ACE, or the spike protein on the SARS-CoV-2 now. This is the open S1 protein up here. And so we can see how that is similar to this over here on this protein. And so I think that was everything I wanted to say about this. Just to quickly review, we are trying to generate this spike protein, the S1 spike protein here, which can then be presented as an antigen. And they are doing that by actually by delivering this mRNA for the S1 into the human body, so that the white blood cells can process this signals to start releasing these interferons. It is translated into this antigen, which is then presented on the cell surface. And then these CD8 and CD4 T cells, which we saw over here, CD4 and CD8 interferons, TNF alphas, and interleukins, so that they can express those, so that these CD8 T cells and CD4 T cells can start expressing those IgGs, which, as I said before, are used to bind onto the viruses and agglutinate them and signal for more mobilization of the immune response. All right, I think that is it for this video. I do make other videos on biochemistry on this channel. I'm making a a playlist on nutrition and metabolism if you are interested in that. And so I hope this was illuminating to you.